Hi, this is Dr. Rosario. This is our second video for Module 7, and the topic of this lesson is microbial nutrition. I'm going to cover two main topics in this video. The first is the nutritional requirements of microorganisms. I will talk about the different types of nutrients that microbes use and how we classify microbes based on their nutrient needs. The second topic in this video will be how microorganisms obtain nutrients, or how the cells take up materials from the external environment. This video is about microbial nutrition. We've discussed in detail so far in this course what constitutes a microbe, so it is useful here to define what we mean by the word nutrition. Nutrition is the process of taking in nutrients and food and using that to support life. All living things require food in some form. Food provides the chemical building blocks of biological molecules. Food is made up of organic material, meaning it contains carbon. Carbon is necessary for building molecules like proteins, DNA, and even membrane phospholipids. Food also provides energy in the form of chemical bonds. This energy can be released and used to drive the work of the cell. We can categorize organisms based on how they use food. These categories are based on the carbon and energy needs of the cell. Remember, carbon is a key element in living things. It forms the backbone of our biological molecules. Some organisms can take carbon up from the environment in the form of carbon dioxide. They use the carbon dioxide to make more complex molecules. This process is known as carbon fixation. So they're using that carbon dioxide to build organic molecules, and the process of capturing carbon dioxide is carbon fixation. Essentially, these organisms make their own food that can be used in a variety of cellular processes. For this reason, these organisms are known as autotrophs. This term means self-feeder. Other organisms, including humans, take up a variety of different organic compounds from the environment, using them as a source of raw material for building new biological molecules. They break down this food and recycle the organic molecules. These organisms are known as heterotrophs. One important molecule used by heterotrophs is glucose, which is pictured on this slide. Remember, autotrophs use carbon dioxide as their carbon source, while heterotrophs use organic molecules as their carbon source. We can classify living things based on their carbon needs into autotrophs or heterotrophs, but we can also classify living things based on their energy needs. Energy is the capacity to do work. All living things must take in energy to maintain life. For example, energy allows cells to move from one place to another. It also makes it possible to build new molecules and to reproduce. All living things need energy. Some organisms participate in photosynthesis. Their primary source of energy is the sun. These organisms capture sunlight energy and store it in the bonds of sugars, using carbon dioxide as a carbon source. Photosynthesis, that word literally comes from photo for sun and synthesis to put together. They're putting together those carbon atoms to build sugars. We already mentioned that an organism that uses carbon dioxide as a carbon source is called an autotroph. Here, because they are using the energy of the sun, they're also called phototrophs. You may see the two terms put together, where an organism that uses carbon dioxide as a carbon source and sunlight energy to link those carbon dioxide molecules together might be called photoautotrophs. Not all organisms can directly use the energy from the sun. Instead, they get energy from the oxidation of chemicals. These organisms are known as chemotrophs. Oxidation is the removal of high-energy electrons. We can further classify chemotrophs based on the source of these electrons. Humans and human pathogens are what are known as chemoorganotrophs. We oxidize organic molecules to release the energy that is stored within those chemical bonds. We depend on other organisms to produce those organic molecules. To be more specific, we are known as chemoorganoheterotrophs because we get the energy from those organic molecules, but we also use those organic molecules as our carbon source. Not all chemotrophs obtain their energy from organic molecules. Some organisms, known as chemolithotrophs, obtain their energy from the oxidation of inorganic chemicals. Most of these organisms are what are known as chemolithoautotrophs, meaning that they get their energy from inorganic molecules, they oxidize those inorganic molecules, but they get their carbon from carbon dioxide. So they're chemolithoautotrophs. This is a picture of a hydrothermal vent at the bottom of the ocean. Chemolithoautotrophs here support the development of a diverse ecosystem that depends on this unique metabolic activity of these organisms to support the entire ecosystem. Whether an organism is a chemoorganoheterotroph or a photoautotroph or any other combination therein, there are 6 elements that are commonly found in biological molecules. They make up 98% of living matter. The first element, of course, is carbon, which forms the backbone of biological molecules. The other elements are hydrogen, nitrogen, oxygen, phosphorus, and sulfur. To remember this, I use the mnemonic SHNOPS. Right? CHNOPS. Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Because organisms must obtain these elements in relatively large amounts from their environment, they are called macronutrients. For chemoheterotrophs, the organic molecules that are regularly consumed in the form of food serve as the main source of these elements. Other elements are also required for living things, but in smaller amounts. These include potassium, sodium, magnesium, chloride, and iron. They are called micronutrients because they are needed in relatively small amounts compared to carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, right? Our macronutrients. Growth factors are organic molecules that organisms must get in their diets because they cannot make them themselves. So they must be obtained as a nutrient. These include things like vitamins, some amino acids, and even some nucleotides used to build DNA and RNA. Growth factor needs will vary from organism to organism. So for example, laboratory strains of E. coli, they don't require any growth factors. So you can just give them a minimal media that just contains those basic elements, those micronutrients and those macronutrients, and then E. coli can grow just fine, just on those elements alone. However, some other organisms like Legionella pneumophila, which causes Legionnaire's disease, require many different growth factors. That particular organism was really difficult to isolate and identify because of its growth needs. Right? And actually, it caused a huge outbreak in the 1970s, but it took like 3 years before they were able to identify that bacterium because they couldn't get it to grow because it was just so finicky. We call those organisms fastidious. Now that we've identified what organisms need to survive, we're going to turn our attention to how they obtain it. We will focus on our chemoheterotrophs because all pathogenic bacteria fall into this category. Chemoheterotrophs, or more specifically chemoorganotrophs, or chemoorganoheterotrophs, get their energy, carbon, and essential nutrients from other organisms. We are in this category, and we eat other living things to survive, right? Whether those living things are plants or animals or some combination of that. Life depends on a food chain where autotrophs, which are mostly photoautotrophs, are the primary producers that all living things rely on. Humans, right, we are predators. We kill other living things in order to get our food, right? So we are predators. Human pathogens are what are known as parasites, right? Parasites get their food from the bodies of other living things while they're alive, right? The bodies of what we call living hosts. Many microorganisms fall into another category known as saprobes. Saprobes are known as decomposers. They feed off of dead and decaying materials. So the food that they eat is already dead before they start to consume it. Microbes get their nutrients from the environment, right? Just like we get our nutrients from the environment. We have to take it into our bodies. Now, microbes don't have complex digestive systems like we do, right? When we eat, we take food in through our mouths, we chew it up, and then that food travels to our stomachs where it gets broken down further, and then it travels through our intestines where the nutrients get absorbed into our bloodstreams. That's not the same for our microorganisms because remember, these are single-celled organisms or maybe just a couple of cells, and they actually have to digest their food outside the cell before they can take that food or those molecules into the cell. So they have to digest their food in the extracellular environment. In order to do that, right, so they're going to digest their food in the extracellular environment. In order to do that, they're going to produce what are known as exoenzymes. Right? Exoenzymes are enzymes that break down larger biological molecules like polysaccharides or proteins into their smaller components. Right? So here you can see those it was probably starch in the image, was broken down into individual glucose molecules. Those individual subunits can be taken up into the cell. So again, these enzymes that are breaking down the food outside the cell are called exoenzymes. And oftentimes there's a link to pathogenicity because those exoenzymes, when they're produced by a pathogen, those exoenzymes can actually cause damage to our tissues, right? And that results in some of the symptoms of diseases that are caused by those pathogens. We can characterize and identify microorganisms in the laboratory by looking for the presence or absence of specific exoenzymes. This is done using specialized growth media. In this example that you're seeing in front of you, you have starch agar, which contains lots and lots of starch, which is a polysaccharide. And we're looking for the presence of an enzyme called amylase. Amylase is an exoenzyme that breaks down starch into individual subunits of sugars. What you can see in this picture is that iodine has been added to the plate. Iodine reacts with starch to produce this purplish black color. So all over this plate you can see this purplish black color except around the area where the bacteria are growing. You see kind of this glowing halo. In that space, all of the starch has been broken down into individual sugars. So the iodine doesn't react to produce that purpley black color. That halo is showing you that this particular organism produces that exoenzyme amylase. There are lots of different kinds of exoenzymes. Each microorganism has their own unique set of tools to extract nutrition from their environment. Here are some examples of other laboratory media that are used to detect the presence of exoenzymes. DNase agar is used to detect nucleases that break down nucleic acids. Tributyrin agar is used to detect the presence of lipase enzymes that break down lipids. And milk agar looks for the presence of proteases that break down proteins. Specifically here, it's the proteins that are found in milk, casein. Obtaining nutrients requires microorganisms to move materials from outside the cell to inside the cell. The opposite, of course, is true for waste products. We want to get them from the inside to out of the cell. Remember, the plasma membrane creates a barrier between the cytoplasm, which is in, and the extracellular environment. The membrane is composed of phospholipids and proteins. The membrane is semi-permeable, meaning that some materials are able to easily cross across it, while others cannot. There are several different ways that materials can move across that membrane. And we're going to talk about those different ways here. Diffusion is the movement of molecules down a concentration gradient, from an area of high concentration to an area of low concentration. There are 3 types of diffusion. We're going to talk about each of these, but they are simple diffusion, facilitated diffusion, and osmosis. The key feature of diffusion is that it does not require an input of energy. So in all 3 of these examples, this is what we call passive transport. It does not require any input of energy. So first we're going to talk about simple diffusion. Simple diffusion occurs when molecules move directly across the membrane. These molecules are usually small, nonpolar substances that can squeeze between the phospholipids that make up that membrane bilayer. Examples of these materials are things like carbon dioxide and oxygen gases. They're small and they're nonpolar. As you can see in this image, The movement occurs from an area of high concentration, in this case outside the membrane. There's lots of these molecules, so that's a high concentration, to inside the cell where there's a very low concentration. This is movement of molecules down their concentration gradient. Facilitated diffusion is like simple diffusion. Here we have molecules moving down their concentration gradient, but the difference is that we require some sort of protein to allow those molecules to move across the membrane. These are usually larger molecules, or sometimes they're charged ions or charged or polar molecules that cannot squeeze between those membrane phospholipids. These proteins are embedded within the membrane and allow a pathway for the molecules to move down their gradient. Again, this is a form of diffusion. The molecules are moving from an area of high concentration to an area of low concentration. The proteins themselves are facilitating the process, which is why we call this facilitated diffusion. Osmosis is the diffusion of water. Right? So whenever you hear the word osmosis, I want you to think water. Water can move via simple diffusion across biological membranes, but it happens very slowly. More frequently, it's going to move through these specialized protein channels that are called aquaporins. Again, just like any form of diffusion, you have water molecules moving from where there's lots of them, or a high concentration of water molecules, to an area where there's a low concentration of water molecules. Now remember, in our biological systems, water is our solvent, right? And usually your other molecules, like your salts and your sugars, are dissolved within that aqueous environment. We will revisit this particular topic in our next lecture video, where we talk about the physical requirements for microbial growth, because water plays a really big role in allowing microorganisms to survive and grow in different environments. So far, we have talked about the movement of molecules down a concentration gradient. But what about a situation where we want to move molecules against a concentration gradient from an area of low concentration to an area of relatively high concentration? This process will require an input of energy and is called active transport. There are three categories of active transport: protein pumps, group translocation, and bulk transport. Protein pumps are used by cells to move materials against their gradient. The hydrolysis of ATP releases energy that powers this process. In this image, you can see that the green triangles are higher in concentration outside the cell than inside, and the purple circles have a higher concentration inside than outside. In this situation, the cell wants to accumulate those purple circles inside while removing the green triangles. Right? We're moving these items against their gradients. You can see here that the pump is open to the outside, and it has pockets that bind the purple circles. It also has a place on the protein that binds ATP. Here it removes one of the phosphate groups from the end of the ATP molecule and attaches it to the protein. This releases energy and will result in a change in the shape of the protein. The phosphorylated protein now opens inward, which allows those purple circles to be released. At the same time, the green triangles are able to bind to that protein pump. Finally, the last step in the process is going to be the removal of that phosphate group, which again releases energy and changes the shape of the protein. It opens back outward, releasing those green triangles into the extracellular environment. This models a system that is commonly found in biology called the sodium-potassium pump. There are lots of different pumps that are found in microorganisms. One example that's relevant to healthcare is that there are protein pumps that actively pump antibiotics out of bacterial cells, creating a type of antimicrobial resistance. The key in this entire process is that it requires energy. Remember, so from the very beginning through the entire cycle, you had the sequential breakdown of ATP, right? You removed the phosphate group from the end of that ATP molecule, you added it to the protein, and then eventually we removed that phosphate group back from the protein to get back to where we started, right? Another form of active transport that occurs in microorganisms is something called group translocation. This is a unique type of energy requiring transport that is used by prokaryotic cells. We're not going to get into the details of it, but I just want you to see that what's happening is that the molecules that are being transported are being chemically modified as they're brought into the cell. And this again allows the cells to accumulate materials in the cytoplasm, right? So again, it's using energy, so this is going to be a form of active transport. The last type of active transport is called bulk transport. This process does not occur in prokaryotic cells, but it does occur in eukaryotes. In bulk transport, the cells use membrane compartments called vesicles to move materials into or even out of the cell. So here we're moving materials using those vesicles, or those little pockets that you see inside the cytoplasm. Phagocytosis is probably the most important form of bulk transport that we're going to talk about in this class. Some of our immune cells use phagocytosis to engulf and destroy pathogens. So that's actually what's pictured on this slide. There are also eukaryotic microorganisms that use phagocytosis to acquire nutrients. Amoebas, for example, use phagocytosis to eat other microorganisms. Well, I'm not going to go into detail here. There are other types of bulk transport, including endocytosis, or what we call receptor-mediated endocytosis. Exocytosis is a form of moving material out of the cell using vesicles. And then penocytosis is a way for cells to take up liquids from their environment. So this is the end of our discussion on microbial nutrition. The last video in this series on microbial growth will focus on the physical conditions necessary to support the lives of our microorganisms.